हेलो हाँ शुरू कर विशांजलि.
Yes, sir. Good morning to everyone. I welcome you on PhD webinar series organized by Microbiology Society India. I welcome to Dr. A.M. Deshmukh sir, All India President of Microbiology Society, and today's speaker, Dr. Dipti Dhere ma'am, and all other participants. I welcome you. Today's speaker, Dr. Dipti Dhere ma'am. Currently, she is working as assistant professor in Department of Microbiology, Dayanand Science College, Latur. Ma'am has 8 years of teaching experience, and also ma'am has attended various conferences and workshop training, and she has achieved first prize for oral presentation in national conference in 2019, and second prize for state level conference in 2023. Also, ma'am has published 11 research articles in national and international journals. Also, ma'am has achieved first prize in Avishkar in 2011 and second in 2018. Second prize in 2018 for Avishkar in category of pure science. Also, ma'am has completed various online courses related to microbiology, such as basic techniques in microbiology, microbial inoculant technology. With permission to our Dr. A.M. Deshmukh sir, President of Microbiology Society, I would like to request ma'am to start her presentation.
Yes, before that, Dipti.
Yes. Yes, sir.
Today there are about 14 participants, and
Yes,
Dr. Prakash Halami from CFTRI Mysore, a very
Yes,
respectable scientist. He has joined today. Yes, and many other. That is, yes, Pramila, Krishna, Kumar, Madhvi, Nikita is there. Then Sharmila Kaulaskar, Sneha Gokte ma'am from Hyderabad. She has also joined. She is Sneha Gokte, a retired professor from Hyderabad. Shivani Pawar, very good. So you can proceed, Dipti, now.
Thank you, Madam, for introducing me, and I'm also thankful to Dr. A.M. Deshmukh sir, President Microbiology Society India, for giving me opportunity to present my research work, and I am also honored for presence of many respected personality for attending this lecture. So, with this brief, I would like to start my presentation. Can you see screen? My screen is visible or not?
No, ma'am.
It is showing here host disabled participant screen sharing. Ma'am, please.
1 minute.
Hello, Dipti.
Yes, sir.
Krishanjali is there. Krishanjali,
Yes, sir.
She has joined by 2 mobile, so you make other mobile as a co-host. You are a
Okay.
host now.
Okay, sir.
Krishanjali, you are host.
Okay.
You make her co-host. Okay.
Okay.
1 minute, sir. Just I want to see that. I don't know that setting.
Just go to list of participant.
Hmm.
Click on Dr. Dipti
Yes, sir.
and make her co-host. Finish.
She's already, sir, a co-host.
2 numbers are there.
Okay. Yes.
So
Yes.
otherwise you make me host. I will make her co-host.
I have done. Okay.
Okay, correct. Now Dipti, you can present your.
Okay, sir. Can you see screen now?
Yes. Yes, ma'am.
Okay. Yes. So the topic of my presentation is the screen is visible. Should I proceed?
Yes, ma'am. Yes, ma'am.
Good morning to all. The topic of my presentation is isolation and screening of carotenoid producing bacteria, process
Pranay,
optimization.
Dipti, wait. Pranay mic is open. Please mute. Pranay.
Sir, I
Please
have
mute.
done that.
Okay, okay, okay.
From my side, I have done.
Proceed. Dipti, proceed.
Yes. So this is my topic. I have completed my research work from Government Institute of Science, Aurangabad, under the guidance of late Dr. Smita Dharmadikari ma'am. So the motivation of my research was that the colors or the synthetic colors are very adverse. Synthetic colors are having very adverse effect on the health. They are allergic, carcinogenic, and cause severe damage to vital organs. On the other hand, the natural colors are very safe due to their biological origin. But the natural colors can be obtained from the plant's microorganism. The plant-derived pigments are having seasonal variation problem related to geographical distribution, and they are not available year around. On the other hand, microbial pigments are independent of seasonal variation, geographical distribution, and we can produce them with ease at a larger level. And that's why I have selected the production of microbial carotenoids. I'm not able to go on next slide. Yes. So the topic of presentation, it is about carotenoids. Carotenoids are yellow, orange, and red pigments synthesized de novo by variety of bacteria, fungi, and plants. The functional nature of carotenoids, that there are more than 700 groups of carotenoids, and carotenoids are light harvesting components in photosynthetic system. They act as a photoprotective in nature. They have antioxidant activity. They are regulator of membrane fluidity, and the main function of carotenoids are they protect cell from the photooxidative damage. The most important thing is that animals cannot synthesize carotenoids, and so their presence in it is due to only dietary intake. We have different types of carotenoids enlisted here. The carotenoids are applied mainly as a colorant, as a feed additive in animal as well as human food. They also act as an antioxidant, anti-tumor activity. They also can be used for the heart disease prevention, precursor of. They also act as a precursor of vitamin A activity and enhancer of in vitro antibody production. And due to lots of application, they can be used in food, medical, pharmaceutical, nutraceutical, and cosmetic industry as a dye and a functional ingredient. So need of carotenoids is briefed here. in this diagram, it is a multifunctional role. Then objective of research. First objective was the isolation and screening of potential carotenoid bacteria. Then identification of bacteria by conventional and advanced molecular techniques. Then optimization of physiological conditions by one at a time approach. Then statistical optimization of media components by using statistical software such as Plackett-Burman design and response surface methods. Then scale-up studies were carried out. Then optimization of extraction process were also carried out. Carotenoid molecule was carried out, and quality control analysis of carotenoid was done, and at last application studies were carried out. Then the methodology of whole research, it was categorized into 3 phases. The first phase, it is related to the isolation, screening and characterization as well as identification of isolate. So the first step was the sample collection. Hypersaline, hyperalkaline, marine and freshwater samples were collected from different these ecosystem, respective ecosystem. They were enriched in nutrient broth. The samples were mainly soil, mud, sediment and water. And isolation was carried out on nutrient agar by serial dilution and spread plate method. Then screening was carried out in 4 stages as a primary, secondary, tertiary, quaternary screening. And after that, potential bacterial isolates was selected. Primary screening was carried out based on the presence of non-diffusible pigment. All participants are requested to mute your mic. I am getting disturbed. So the primary screening was done basically depending on the presence of pigment, that is yellow, red, orange pigment, which are non-diffusible in nature. Secondary screening was based on the sodium chloride tolerance and the tolerance to pH, that is 8 to 12. Tertiary screening was based on the spectral characters of the pigment as well as degree of pigmentation. Degree of pigmentation was the ratio of pigment produced to the biomass produced. Quaternary screening was done based on the free radical scavenging activity, that is antioxidant activity, which was performed by the DPPH method, and the selection of potential bacterial isolate after all these 4 screening was done based on the pigment characters, degree of pigmentation and the possible outcome on the commercialization. Then phenotypic characterization was done where morphological colony characters, motility, microscopic imaging was done. Biochemical characterization was also carried out by conventional techniques, and the advanced identification was done by the 16S rRNA sequence analysis and phylogenetic reconstruction. So these are the findings of the phase first. Here you can see the samples collected. Total 88 samples were collected from the lunar crater Buldhana, marine sample from various marine sites of India, then Salim Ali Lake, Chhatrapati Sambhajinagar, Harsul Lake, Panchakki, and Hatala Lake. So the physiochemical properties of the collected water sample were also analyzed. Then samples were distributed depending on the types of sample, that is sediment, soil, water, and mud. According to this, it is presented here. Then the percentage of sample recovered from different sources. Here you can see the maximum samples were recovered, that is 31% from the lunar crater. Then primary screening was done depending on the presence of non-diffusible pigment, that is yellow, red, and orange pigment. And here you can see the we got the 9 these much isolate from the different samples. Here the distribution of bacteria is shown depending on the source of collection. Then distribution of pigment and non-pigmented bacteria is shown here. 17% isolates were recovered only from the non-pigmented one, and the pigmented isolate, in which depending on the presence of pigment or the color of pigment, we have distributed them. So we got the maximum yellow orange isolate and minimum red isolate. So Then secondary screening was carried out depending on the tolerance to pH and sodium chloride. It was done by simply streaking on the nutrient agar containing different pH and different salt concentration separately. And we the most tolerant isolates which are having carotenoid production ability, they were selected for the next screening. Then tertiary screening was done depending on the extraction and the spectral studies of pigment and degree of pigmentation. And here you can see the SL2 isolate show the maximum carotenoid production, that is maximum degree of pigmentation was shown, that is pigment produced to the biomass produced ratio by SL2. And the predicted pigment was astaxanthin, which was predicted depending on its lambda max, that is absorption maximum, and that's why it was selected as the next. It was selected for the next screening level. Then quaternary screening. For quaternary screening, these isolates were selected from the tertiary screening, which were carried out by the antioxidant activity. During this, antioxidant activity was carried out by the DPPH method, and it was found that the SL4 star and SL2 isolate shown the highest free radical scavenging activity, out of which, when we analyze the chemical nature of carotenoid by the pharmacological test to determine the presence of Pauling chain in the carotenoid in that pigment, it was found that the SL4 pigment, it is non-carotenoid in nature, and that's why obviously we have selected this SL2 strain, which shown the presence of Pauling chain in the pigment extract and high free radical scavenging activity, and that's why we have selected it for the next level of studies. So we have also determined the color code of the bacterial pellet by using the Minsil color code. We have also characterize the isolate SL2 depending on its morphological and morphological characteristics. So here on plate you can see the beautiful pigmentation or the carotenoid production by the SL2 pigment on the plate, that is on nutrient agar. Then biochemical characters of isolate SL2 was also studied, where we have studied the utilization of carbon source, substrate utilization, and enzyme activity studies were also studied. And here you can see this isolate, it is only positive only for 3 types of activity, that is gelatinase, oxidase, and the catalase. Other activities were not shown by this isolate. Then the biochemical characters, depending on the biochemical characters, the SL2 isolate, it was characterized as a Paracoccus species by using Burgess manual, and after that the strain was sent to the 16S rRNA nucleotide sequence analysis to the Agarkar Research Institute, from where we get the partial sequence of 16S rRNA and the phylogenetic tree. And depending on that, the SL2 isolate was showing near about same characters with the Paracoccus marinus and the Paracoccus bibensis. And that's why we have made the comparative studies of isolate SL2 with the Paracoccus bibensis and the Paracoccus marinus, and we found that the isolate SL2, it shows the maximum similarities with the Paracoccus bibensis JLT1284T, and that's why it was concluded as isolate SL2, it is a Paracoccus bibensis than the Paracoccus marinus. Then these are some photographic presentation of the results. Then conclusion of the phase first, that from 88 samples, 620 isolates were recovered, out of which 106 were pigmented isolate. On screening, bacterial isolate SL2 was selected as a potential carotenoid producer. Highlights of the SL2 isolate, that is Paracoccus species, it is tolerant to 6 to 12 pH. It is tolerant to 1 to 6 sodium chloride sodium chloride concentration. It produces a bright orange non-diffusible pigment showing absorption maxima at 473 nanometer in and it is chemically Pauling in nature. It shows 53% radical scavenging activity, which was estimated by DPPH assay. And the isolate finally was identified as Paracoccus bibuensis strain SL2, and sequence was deposited to NCBI gene bank with the accession number KY129665. Then in phase 2, we have carried out the optimization studies, scale-up studies, and the production of the carotenoid. The methodology includes optimization of physiological parameters, which includes pH, optimization of pH, aeration and agitation conditions, incubation of temperature, incubation of time, effect of different wavelength of light on carotenoid production, then standardization of media components by single factor analysis, that is carbon source, nitrogen source, and sodium chloride concentration. These media components were optimized by one single factor analysis. And after that, the optimization of media components was done by statistical optimization, where we have used Plackett-Burman design and response surface technique. The Plackett-Burman design for which we have used the Minitab 14, and for response surface technique we have used the design expert 10. Then for optimization studies, it was very essential to select a basal media for carrying out the optimization study, and that's why, as we have enriched and isolated strain from the nutrient broth, we have used the simplest media for the enrichment for the optimization studies, and it was found that the peptone water was the best basal media for the optimization study, as it was showing the highest carotenoid accumulation. Then optimization of pH was carried out, where we found that the optimum pH for carotenoid accumulation was 9.5, and which highlights the alkalotolerant nature of Paracoccus bibuensis. The every optimization studies was tested statistically by using pair t-test for its significance, where p-value less than 0.05 was observed, which proves the significance of the result. Then optimization of aeration and agitation conditions were carried out, as we know that the Paracoccus bibuensis, it is aerobic in nature, and that's why we have optimized the aeration agitation condition, and it was found that 120 rpm, it was the optimum aeration condition for the carotenoid accumulation. After that, there was no improvement in the carotenoid accumulation. Then optimization of incubation time was also carried out. It was found that the carotenoid pigments are primary metabolites. They are produced mainly in the primary growth phase, that is logarithmic growth phase, and initial stationary phase, and that's why the maximum carotenoid production was found during 12 to 72 hours, and after that there was constant reading about the carotenoid accumulation. Then incubation temperature was also optimized, and 30 degrees centigrade, it was found the optimum incubation temperature for the carotenoid accumulation. The effect of light on carotenoid production was studied. As we know that the carotenoid production, carotenoid production, it is due to the in response to the light, and that's why we have studied the effect of different wavelength of light on carotenoid production, and we found that the maximum carotenoid accumulation, it was in response to the sunlight and white light, while the red light, it was adversely affecting or showing the minimum type of carotenoid production. For this we have carried out the jugaad. We have wrapped the flask with the colored paper and incubated that in white light at 30 degrees centigrade in a walk-in cooler, and after that we have carried out extraction studies and studied the pigment produced, and we found that the white light and sunlight, it was the most influencing parameter for the carotenoid production. Then optimization of carbon source was carried out. The use of simplest sugar was the aim for the carotenoid production, to make the process more economic. We have tried different sugars for the carotenoid production and found that the arabinose and the glucose, they were the showing highest carotenoid production, as the arabinose sugar, it is very costly, and to make the production more economic, we have selected glucose as a sugar source for the next level of carotenoid studies. Then the combination of different sugars were also studied. In that also we have seen that the maximum carotenoid accumulation was with the glucose. Then the different concentration of glucose were studied, and it was found that the 1% glucose, it was optimum concentration for the maximum accumulation of the carotenoid. 0.5% glucose gives the highest yield of carotenoid, and hence the selected for the next level of optimization studies. So here you can see the pictures. Then optimization of nitrogen source for carotenoid production was carried out, where we have screened both the organic and inorganic nitrogen sources. Any kind of carotenoid production was not observed when we have added inorganic nitrogen sources, while there was carotenoid production was observed in response to the organic carotenoid production, as it is due to the complex nitrogen source, as the organic nitrogen sources are complex in nature, and in response to that, there is a generation of peroxide radicals, in response to which carotenoids are synthesized, and that's why we get the maximum carotenoid accumulation in response to the organic sources. In that also, the meat extract given the highest carotenoid accumulation. Then when we use meat extract as a sole nitrogen source, before that we have optimized nitrogen sources with the different combination, and in that also we got the highest carotenoid concentration with the meat extract. Then we have optimized different concentration of meat extract, and it was found that 1.5% meat extract was sufficient or optimum concentration for the carotenoid production. Then sodium chloride concentration was also optimized, and it was found that 1% sodium chloride concentration was the optimum concentration for the carotenoid accumulation. Though the organism was halotolerant, but the maximum carotenoid concentration was, or the accumulation was found with the 1% carotenoid. Then optimization of media components was done by statistical approach, where we have used the Plackett-Berman design, and after that it was found that the single nutrient factor, that is meat extract, it was affecting significantly on the carotenoid production. So the Pareto chart shows the results. Then the response surface methodology was carried out by the central composite design, where we have used Design Expert 10 software. In that also we found that the meat extract concentration, that is 1.5% meat extract, was the optimum for the carotenoid. Then CCD, that is the central composite design, was carried out for the single factor analysis, because wheat extract was the only factor that we found significant for the carotenoid production, and it was found that the meat extract was the best nitrogen source and the significant parameter for the carotenoid production. So here the comparative analysis between the nutrient broth peptone water optimized media and the carotenoid statistically optimized media for the carotenoid production was done. Then scale-up studies were carried out. The large scale, in that the optimization of inoculum size was done. The optimization of inoculum age also carried out, and after that it was found that the 1% inoculum was the optimum for the carotenoid production, and the inoculum with the age 48 hours was the optimum for the carotenoid production. Then the optimization of ratio of flask capacity to the flask volume was carried out to know the best ratio for the media transfer to the volume, and it was found that the 100 ml flask was sufficient for the carotenoid production, and media volume, it was the 25 ml. So the 25 ml in 100 ml flask, which gives the ratio 1:4, which indicates the optimum transfer transfer of nutrients, growth factors, and oxygen, which helps to the scale up of process. Then scale up of carotenoid production was done at flask level from 100 ml to the 1000 ml. We have carried out the carotenoid production by increasing the scale but keeping the ratio constant, and it was found that the carotenoid production was not adversely affected by the increase in the scale of production, and the general behavior of Paracopus bibasis was found independent of scale of production, which these findings are adding good values to the research studies and will positively impact on the fermentation at industrial level. Then carotenoid production by batch and fed batch fermentation. The carotenoid production by batch and by batch fermentation was carried out, and it was found that the positive effect of glucose was found on biomass production than the carotenoid production. The maximum carotenoid accumulation was found mainly after 72 hours, that is 1.05 milligram per liter. The carotenoid production was carried out also by fed batch fermentation, and it was found that after 2 feeding of the same media during the fed batch fermentation, the accumulation of carotenoid was improved as compared to the fed batch fermentation. That's why the fed batch fermentation was used at the next level of optimization, that is scale-up studies, where we have used a tank fermenter of 5 liter capacity for the studies, where we have carried out the fed batch fermentation, and exponential growth phase was reported at initial 96 hours, which was declined slowly up to the 180 hours. The maximum carotenoid accumulation was found 5.5 milligram per liter with the fed batch, and that's why this fermentation studies were concluded. The conclusion of phase 2 was that the pH 9.5, aeration agitation condition 120 rpm, incubation period 72 hours, incubation temperature 30 degrees centigrade. The carotenoid production was positively influenced by sunlight and plain white light, optimized nutritional component by single factor analysis and statistical analysis by Plackett-Berman and response surface methodology. The optimized media for carotenoid production was glucose 0.5%, wheat extract 1.5%, and 1% sodium chloride concentration. The optimum inoculum size was 1%, inoculum age was 48%, the ratio of media volume to flask was 1:4. The carotenoid production was not adversely affected by increasing the scale of production. The carotenoid production was carried out at fermenter level, and the yield was found 2.77 milligram per liter at shake flask level. The large scale production was done using 5 liter capacity stirred tank fermenter with the 5.5 milligram per liter of yield. Then phase 3 studies includes optimization of extraction, then extraction of carotenoids, and quality control analysis. In this, first extraction methods were optimized. Then quality control analysis was carried out, where we have studied toxicity study, acute toxicity and cytotoxicity. Then stability of carotenoids to the pH, temperature, and the light. Then we have analyzed or characterized the pigment by doing different methods. That is pigment. We have separated first pigment depending on the polar and non-polar nature of pigment. Then chromatographic fractionation and TLC was carried out. Then identification of carotenoid was done by spectrophotometric method and chemical test. I'm extremely sorry. Here there is no electricity. I will switch on to the mobile if it switches off. Okay. Then the carotenoid analysis was done lastly by the. After that, application of carotenoids was done. Was studied as an antioxidant, as a feed, fish feed applicant, as a colorant to the different things. So the phase 3 includes the optimization of carotenoid extraction parameter, in which the method A, which includes the freezing thawing with the acetone methanol concentration, that is the ratio 1:1, given the relatively higher yield, that is 12.998 microgram than the method B, C, and D. Each finding were statistically tested with the pair t-test. Then optimization of organic solvent was done, and it was found that acetone, it was extracting highest carotenoid. It was recovering highest carotenoid. Then combination of different organic solvent were also tried. In that also we found that acetone as a sole organic solvent for the extraction of the carotenoid. Then effect of acetone concentration was studied further, in which we found that the absolute acetone, that is with 100% given the highest carotenoid extraction. Then carotenoid extraction parameters, that is temperature and time, were studied. We have extracted carotenoid for the different temperature, and it was found that at 60 degrees centigrade, maximum carotenoids were extracted from the cell. The higher temperature may be destabilize membrane lipids, which interfere in the extraction of carotenoids, and that's why the 60 degrees centigrade, it was the optimum carotenoid extraction temperature. And at 25 degrees centigrade, the highest carotenoid extraction was done. Then separation of pigment was done depending on its polar and non-polar nature. In this, we have removed major contaminating lipid, separated pigment based on the polarity. The method was useful for the large preparatory work and predicted probable group of the pigment. Here we can see the pigment was accumulated in the hypophase and the epiphase. The epiphase is of the chloroform and the petroleum ether, and it is of acetone and the methanol, which are the polar solvent. And the presence of carotenoid pigment in the polar solvent indicates the xanthopyl group pigment of the carotenoids. Then chromatographic fractionation of carotenoid was carried out, where we have carried out pigment purification by chromatographic techniques, and the separation of the carotenoid on the TLC, and the RF value was determined. The several functional fraction of open column chromatography was obtained. Spectrophotometry at 480 nanometer was used to group this fraction into 5. The purified fraction of spectrophotometric analysis showed the absorption maxima at 480 nanometer. Then characterization of pigment was done depending on spectrophotometric characterization, and the methanol extract showing the 480 nanometer as a absorption maxima with a broad shoulder region. It was the characteristic feature of the xanthopyl group pigment, and it also shows the presence of polycystin, that is 10 or 11 presence of double bonds. Then confirmation of chemical nature was done. Pharmacological test for detection of carotenoid was done, where we have taken the dry extract, or the extract from the dry pellet of the organism into the chloroform, and that was mixed with the concentrated H2SO4. On that interface, if the blue color ring it was found, the it indicates the presence of polyene group, and the presence of polyene group in the pigment, it confirms the carotenoid nature of the pigment. Then FTIR analysis was carried out. In that we found that the OH keto group and the carbon-carbon chain, it was optimized. Then HPLC analysis was carried out, where we found that the presence of trans-astaxanthin pigment, it was compared with the HPLC spectra of the standard carotenoid astaxanthin. It is the predicted structure, that is 2D structure and the 3D structure of the pigment. Then we have carried out the acute toxicity study. It was carried out on the goldfish. Goldfish was fed with the carotenoid mixed into their feed, and after feeding to the goldfish for 96 hours, there was no any kind of adverse effect, that is asthenia, imbalance, spiral swimming, and the collapse to the bottom to the aquarium was not observed with the fish, and hence carotenoid extract was concluded as non-toxic. The next level of toxicity was studied as a cytotoxicity on the human blood lymphocyte by the MTT assay. Then sterility test was also carried out, and after that stability studies of the carotenoid pigment was carried out, and it was found that carotenoid pigment, it was more stable to the 9 pH, and it retained its stability to the wide range of pH, and hence we can use it safely without major loss of its activity. The stability of carotenoid to the temperature was also observed, and it was stable up to the 20 degrees centigrade. After that, as temperature increases, the stability was decreased. The stability of carotenoid was decreased with the sunlight, while it was more stable to the white light, followed by UV light and the dark condition. Then application of carotenoid as an antioxidant was studied by the DPPH method, where we have used 0.1 millimolar DPPH in methanol, and this is the protocol for the antioxidant activity, and where we found that IC50, that is 4.6 microgram per ml, IC50 value was recovered. The IC50 was estimated by DPPH method. IC50 was calculated to know the potency of carotenoid as an antioxidant. Pigment was estimated, IC50 was estimated as 4.6 microgram per ml. The PHT was used as a positive control with IC50, that is 7.2 microgram per ml. IC50 was observed. The IC50 value of standard carotenoid was reported as 5.2 microgram per ml. Then application of carotenoid as a feed supplement was studied in fish, goldfish, that is Carices auratus, where by using this formula, we have prepared the fish feed pellets supplemented with the carotenoids. Then blood parameters of the goldfish was carried out, and the other parameters, that is specific growth rate, percent mortality, average weight gain, all these parameters were studied. The fish fed with the extracted pool carotenoid were brightest in skin color, and the fish fed with the control were brightest in skin color. These two segments were judged with the evaluator group, and the 72% were agreed with the first statement, and 28% evaluators were agreed with the second statement. Then application of bacterial carotenoids was done with the for the different preparation of different goods, that is eco-friendly candles, carotenoid supplemented soap, carotenoid supplemented cosmetic. Then its effect on skin was observed. Then supplemented lip balms as a cloth colorant, as the preparation of Indian food, that is preparation of crude oil. Then carotenoid supplemented olive oil. And these are the some findings. These are the test feed and the control feed, and these are the control fish and test feed at the end of experiment. So then comparative analysis of research studies where with the previous reported findings were carried out, and it was found that the Paracoccus SL2, though it was previously reported as a Paracoccus bibensis and Paracoccus carotenificans, the maximum carotenoid accumulation was with the SL2, carotenoid SL2, and it was extensively studied up to its maximum parameters than the reported findings. It is the, I think it is the first Paracoccus species reported, which was extensively studied up to this level. Then conclusion of phase 3, the extracted carotenoid has structural similarities with the xanthophyll group carotenoid. That is, it was concluded as astaxanthin, depending on all characterization studies, was the carotenoid molecule was found to safe, non-toxic, as well as stable to change in pH and temperature. The carotenoid extracted in recent studies have highest antioxidant activity, due to which it can be used as a molecule to prevent onset of many lifestyle-related diseases and age-related degeneration. In existing scenario, most of the food colorants used are chemically synthesized, which are harmful. Bacterial carotenoids are natural pigment, can be used both as a color and functional molecule. The bacterial carotenoids produced can be safely used as a feed supplement and as a food additive due to its natural origin, and hence it can be used in food industry and safely in aquaculture. The cosmetic supplementation by carotenoid can reduce aging and the skin problems. Supplementation of carotenoids in oil can prevent its rancidity and improve the healthiness of oil. At last, bacterial carotenoids can be efficiently rule out synthetic food colorants which are not healthy. So the future perspective includes to use some more advanced technique for the structural elucidation of the extracted molecule, then determination of anti-cancerous activity of the carotenoid on the cancer cell line, and the stain improvement studies and the genetic analysis. Then these are some presentation and publication with this research studies. Then this research studies were presented in many conferences. These are the abstract published. For these research studies, I acknowledge these team members, that is for 16S rRNA sequence analysis for large-scale fermentation studies. Then cytotoxicity.
Krishnanjali, I think speaker is not there. It was showing a low battery in our mobile. Yes, you can ask.
So I have switched to the mobile. So with this brief, I
You can
conclude
put
my
up your
presentation.
video. Yeah, go ahead.
If there are any questions, you can ask. Hello, ma'am.
Well, hello.
Yes, sir.
Dr. Dipti, you have presented very well presentation. Thanks for that. I have few queries regarding this presentation.
Yes, sir.
Like you have searched literature a lot. What do you think? Which source is best for carotenoid production?
Microbial source is always good for the carotenoid production, sir.
No. Which?
Habitat. Sir wants to know which habitat. Is it not, sir?
No. The genus. I'm interested in knowing the genus.
Microorganism, which genus?
Yes, sir. In bacterial species, the Paracoccus species, it was studied by me at the first time with all these studies. Other workers, they have only done the studies up to the isolation, screening, and characterization of pigment up to 2019. After that, I have not gone through the literature, but the maximum findings were with the yeast species later on, which I found that the Rhodotorula species, it was good. It was found good for the carotenoid production. It was yeast.
Okay, fine. And you also use the DPPH assay for screening purpose for carotenoid production. Is there any relation between the DPPH and the carotenoid production?
Yes, sir. We were with the motto that the pigment which shows the maximum antioxidant activity has to be commercialized, and that's why we have used that one of the screening parameters.
Okay, fine.
Because
you very
it can be used as a functional molecule.
Okay, thank you.
Thank you, sir.
Dr. Dipti, can you hear me?
Yes, sir.
Compared to fungal source, how is the bacterial source? Because I see even you have to grow till 72 hours, right? Which looks
Yes.
quite a longer duration for a carotenoid production, and we see even in the fungus, somewhere at 70-80 hours, actually we get the good carotenoid production. The one which you were actually referring, similar work has
Yes,
been
sir,
carried
but
out in Rhodotorula,
Yes, sir.
Rhodotorula gracilis.
Yes,
Another
sir.
thing is, if you do the Rhodotorula gracilis, same fungus can produce a different color.
Yes, sir.
If you do the mutation,
Many
or if you
different
change the
carotenoids were reported with the Rhodotorula.
yes. So did you come across with this paracoccus also? Similar
No,
observation?
sir. In research findings, not extensive studies, but reported that it is a carotenoid producing strain. Paracoccus marinus and Paracoccus bibuensis, these two strains were reported previously for the carotenoid production.
Paracoccus is pathogenic.
No, sir.
You check hemolytic lecithinase activity, you have checked, right? Cytotoxicity also you have checked?
Yes, sir. We have carried out cytotoxicity activity and active toxicity also of the extracted
So
pigment,
you-
not the bacteria.
No, not for the bacteria.
No, sir. We have carried out it only for the extracted pigment.
Only for the pigment.
Yes, sir.
How did you initially screen these cultures? Is it a random screening or you go with a specific screening? Selective screening, I mean.
We have carried out selective screening at 4 level, that is primary, secondary, tertiary, and quaternary screening. In primary screening, we have screened based on the colors. Secondary screening
Okay.
based on the tolerance to pH and sodium chloride. Tertiary screening was based on the its absorption maxima, that is spectral characteristics which are similar with the carotenoid pigments, as well as the degree of pigmentation, which is the ratio of pigment produced to the biomass produced. And quaternary screening was based on the antioxidant activity which was done by the TPPH method.
Now, how did you come to the conclusion that SL12 only is the best one? You didn't have any other culture also?
Yes, sir. Yes, SL2, it was shown the maximum carotenoid production with the lowest biomass, that is shown highest degree of pigmentation. It showed the lambda max at 480 nanometer in the acetone, which confirms the the carotenoid can be predicted as a astaxanthin from previously reported findings, and it also showed 53% of DPPH activity or the antioxidant activity by the DPPH method, and that's why out of all isolate we have finalized this, because the main parameter for finalizing this SL2 isolate was its high production and the nature of pigment, that is presence of astaxanthin. This astaxanthin, it is a very functional molecule at a commercial level and can be used for the different applications.
5.5 milligram per liter. This is the highest yield you report, or any other workers have also shown the similar yield in the fermentation or in the scale-up study?
Sir, I'm not getting your question. Voice is not clear.
5.5 milligram per liter yield of carotenoid you have demonstrated, right?
Yes, sir.
Is it the highest yield which is shown till now, or similar study has been reported? Can you hear me?
It was not reported with the bacteria. Similar studies or better production may be reported with the fungal species, but not with the bacterial species, sir.
Okay, very good, very good. Reduction in aging, you said carotenoid can be used in reduction, but you have concluded that this carotenoid pigment can be used to reduce the aging, but you have not done any experiment, no?
No, sir. No.
Why did you conclude then that this can be used for anti-aging?
hello, sir.
That is you have just you propose, right? That is your opinion, right?
Yes,
Can you
sir.
hear me?
No, sir, your voice is not clear.
Can you hear me now?
Yes, sir.
You said that this carotenoid can be used to reduce the aging, right?
Reduced in?
Aging, reduction
Not,
in aging. You
yes,
have
yes.
you have
Reduction in
described.
aging. No, sir.
Okay.
No, sir, we have not tried. It is only reported findings in the literature.
In literature. Okay. And then
Yes,
you
sir.
did not do
But it
any
can be used from
antibacterial
future
activity.
perspective. We are going to do those studies.
Exactly, that is your actually is interesting that, you know. And
Yes,
you can actually do again the antibacterial activity because most
yes,
of this
sir.
carotenoid has a
Yes,
very good
sir.
antimicrobial activity.
We have carried out that. Here we have not
Yeah.
reported in the in our finding, but we did not find any kind of good antimicrobial activity with this carotenoid, because the carotenoid
Okay.
activity by the methanol and the carotenoid by activity by the carotenoid extracted in the methanol was showing the same zone of inhibition. That may be due to the methanol. So we have not studied that in to the further level.
The last question is tell about the photostability, light stability of
Yes,
this
sir.
carotenoid.
They are stable to the white light, because carotenoid
Okay.
pigments are produced in response to the light, and that's why
Exactly.
as they are protective in nature, they are more stable in white light. But they may be damaged. The extracted pigment was damaged by the sunlight, but not the pigment associated with the cells.
Very good study. Congratulations.
Thank you, sir.
Hello, ma'am.
Yes, yes, sir. Yes, ma'am.
I want to know whether on large scale is it possible to produce this pigment?
Yes, we have produced it up to the 5-liter capacity, and it is possible to commercialize this pigment by its large scale production. But before that, many studies related to the fermenter pilot scale studies are required. That may be somewhat different. Parameters may be somewhat different.
But ma'am, means I feel or I think it is very tedious now. Means to apply in food or in apply in cosmetics. Is it possible to extract such large amount of pigment and
Yes.
add it in food?
Yes. Extraction process is very easy, because for extraction we have to only grow the bacteria with optimum condition. Then we have to separate the pellet, and that pellet we have to treat with the acetone by freezing thawing method at 60 degrees centigrade. Within 25 minutes, we can take that extract outside, which has the extracted pigment, and after evaporation of that pigment, we can take it for the different application studies after the purification, by chromatographic purification and all. The purification level, it is also dependent on the use of its level. If you want to use that as a feed additive, then we have to purify it at a with the stringent factors.
Hello, ma'am.
Hello.
Yes.
Okay, there are some questions from other participants too.
Yes.
Amrish Sindegi sir want to ask a question.
Yes.
Sir, you may ask question directly to ma'am.
Hello, madam.
Yes, sir.
Can you hear, ma'am?
Yes, sir. Yes, sir.
Madam, I need to ask a question. Like during this one, biopigment production with this one solid state fermentation.
Yes, sir.
Solid state fermentation. So, which source will be good? Like agriculture waste or fish processing waste, or any other waste you can get for the scale up of bioequipment production, scaling up?
Sir, see, we have carried out the biochemical studies in that we did not find any kind of enzyme activity except oxidase, gelatinase, and the catalase activity. And that's why we have not tried any kind of agro residues. This kind of studies may be possible with the fungal strains which have the ability to produce extracellular enzymes. In that case, you can use the agro residues as a substrate for the carotenoid production. Such studies are reported pure. I have not done,
Yes,
not
ma'am.
done such studies with
No, madam. I have gone through the very good papers and all elsewhere and all. They have used agriculture waste and this one crab cell waste or fish processing waste.
yes, sir.
So there it will be yield will be high because I'm not talking about your strain. I'm not
Yes,
talking about your strain, but the strain which they have isolated from the soil and water, the wastewater or effluent or sludge. So they have end up with a good this one yield in the agriculture waste or fish processing waste, whatever the waste generated after the fish processing.
Yes,
And
sir.
crab cell, especially I had some papers on crab cell waste like that will be good for the fermentation to carry out,
Sir,
and you
which
can
species,
get the
fungal or bacterial?
bacterial many bacteria they have worked on that, many bacteria. I had
It-
gone through nearly 100 papers.
Adding my knowledge.
I got means I gone through many papers, nearly 100 papers. So that I'm just searching out the good stores. Corn cob is a good right now, and we are planning for the crab cell waste to scale up the pigment production. So we are working with some engineering students, biochemical engineering and chemical engineering. So we are trying at our level best.
Okay, sir. All the best, sir. You have also added to my knowledge. In our future studies, we are trying to optimize the carotenoid production by using cheapest source, agro residues or other residues.
Yeah, because bioprocess, this one fish processing also now they are going to waste and they will dump here and there. So that
Yes,
we
sir.
can use for this one and corn and agriculture waste. And even say egg cell we can inculcate for the production of this one egg cell.
Yes, sir. If organism has ability to utilize those substrate, definitely it can be used for the production.
Okay, okay, madam. So, sure, I
Thank you,
will
ma'am.
have you.
Uh,
I will have your contact number, ma'am, and your email ID, so that it will be benefit for me.
Okay,
And
sir.
how
I will be pleased, sir.
yeah, we'll have the interaction in between our study.
Okay,
That,
sir.
thank you, ma'am. Thank you very much.
Hello. Hello,
Yes,
ma'am.
yes,
There are other two participants. They also have questions, but as we have time limit, so we are going to take that questions hurriedly. And participants are requested to take email ID or contact number of ma'am, and you may have discussion on this topic because we have time limit. That's why.
ma'am, you can share my email ID and
Yes,
so that
you can put it in that chat box also, ma'am.
okay, okay.
So the another participant having question is Dr. Sneha, ma'am. Ma'am, you may ask question.
It was a nice presentation. I have two questions. You have said that white light stabilizes the carotene production. But when you compare with carrot, which is underground, how do you explain the comparison of carrot producing carotenoid and your bacterium producing carotenoid? Is there any difference in the kind of enzymes?
Yes, ma'am, that may be different. The bacteria generally produce.
No. Have you come across anything that what are the enzymes required
for carotenoid production? So yes, ma'am, we have studied the metabolic pathway for the carotenoid production, and I think MAPE pathway it is
Is it different in carrot?
It may be. I have not gone through the pathway from the carrot, ma'am.
Yeah. Second question is, carrot can give you lot of pigment. Then why do you go for bacterial pigments, which are very scanty and tedious process? So how will it become economically feasible? You have
Ma'am.
done only 5-liter studies, not even the pilot scale.
No, ma'am. Yes.
And
Ma'am, generally,
carrot is available in plenty. It gives lot more biomass and production of pigment. So how do you justify production by bacteria?
ma'am, generally carotenoid that we take from the carrot, we do not take it regularly. And when I have
What
studied
do you mean by regularly?
regularly means daily or
Why?
on compulsory basis. Generally, the most of Indian population do not
No,
go.
if you are giving a concentrate obtained from carrot, my question is not carrot whether you take it daily basis. My question is, carrot will give you lot more pigment in terms of biomass weight as compared to your bacterium, and bacteria will take very tedious procedure to produce, I mean, your fermentation technology. So how do you justify that production by bacterial population is economically viable? I'm not saying whether our population eats carrot or not.
Ma'am, bacterial carotenoid pigment production and extraction process, we have studied the economics of that fermentation also.
No.
And we have
But
find
carrot extraction, have you seen?
Carrot is not
Research
year-round
papers.
available. It is not year-round available. It is seasonal. It is depend on the season and variation also.
My question
on the seasonal variation.
Hello. No, Deepti, hello. My question is, have you seen in literature carrot to be extracted or used for production of carotene?
No, ma'am, no. I have not. As a microbiologist, I have studied the microbial sources for the carotenoid production, because the carotenoid was aimed to use as a functional molecule, and to improve its bioequivalence, the portion of carotenoid should be absorbed in the lower intestine. It should be with a proper amount, and the carotenoid extract that we take in the form of carrot juice or in any other form which is plant based, that extraction of that carrot, it is a very tedious method, ma'am.
And bacterial carotenoid, does it dissolve in water easily?
No, ma'am. Carotenoid pigments, they are lipophilic in nature. If you take carrot along with the any oil, then its absorption in lower intestine increases. It is that's why we have the recipe like gajar ka halwa, which has the carotenoid along with the ghee, which improves the absorption of carotenoid in the intestine.
Yeah, good. Thank you.
Thank you, ma'am.
Hello, ma'am. There are few questions, but I think we have time limit, so you share your contact number.
Hello, Dr. Aroskar here. Dr. Aroskar here.
Hello, sir.
Hello. Dr. Aroskar here from industry.
बोलिए.
अच्छा,
We can hear you, sir.
Yeah, Dr. Dipti, can you
Yes,
hear
sir.
me?
Yes, sir.
Yeah, already I'm from industry. 40 years working in fermentation, pharma, nutraceutical industry.
Yes, sir.
Like Cipla, Lupin, RPG, Wire, Fermentabite. And
Yes.
this beta carotene is already been manufactured in Europe and US. by
Yes, sir.
fermentation technology.
Yes, sir.
And but the yields are very high. The yields are around about 10 gram per liter, where it is economical. 10
Yes,
gram per liter.
yes, sir.
So beta carotene is already they don't extract from carrots now, but it is done by fermentation technology.
Yes, sir.
Okay, thank you. You can give your contact to me,
Yes, sir. I have posted it
email
in
address.
chat box.
Okay,
Thank you, sir.
okay.
Hello.
Okay. Hello.
Hello,
Yes,
can
ma'am.
hello, Dr. Gupti.
Yes, ma'am.
Actually, I have one question. You are telling that it is halophilic in nature, your bacteria,
Ma'am, I have
isn't
said
it?
that it is halo tolerant, not halophilic. The strain, it was originally isolated from the alkalophilic environment of lunar crater. So it was found as a halo tolerant,
Okay.
not the halophilic.
Okay, if it is halotolerant, how it is helpful actually? So it can tolerate up to what percentage of your salt concentration, and how it is helpful in the production of carotenoid? Like in
No,
the media, you want to use other.
it was alkalo tolerant. We have used screening criteria as salt tolerant because the carotenoid pigments are produced in response to the stress condition. The high salt concentration, it is also a stressful condition to the microorganism. And that's why here we have checked the salt tolerance of the bacteria, and we have found that it is a salt tolerant. That means it can deal with the stressful condition as well as it can produce the carotenoid. To know the stubborn
But
nature of
up to
organism,
it
we
was
have-
only 1%, no?
yes, ma'am. It is tolerant, but for carotenoid production, it was optimum, that is 1%. It's
Okay.
tolerant up to the 6%.
Yeah, I understood. Next is now, why don't you use any control actually?
Yes, ma'am. We have used control.
What did you use?
Control without salt or control.
No, not like that. As a carotenoid producer, you have used that. Okay, your bacteria is producing carotenoid as a commercially available carotenoids.
Yes,
Have
ma'am.
you
We have
used
used astaxanthin?
commercially available
Yes,
carotenoid?
yes, ma'am. Astaxanthin by Sigma we have used as a
Okay.
control.
And whether it is very effective, cost effective?
Yes, ma'am. It is cost effective. I think I have. Yes, we get the commercially available. That is 132.5 rupees per milligram. It is the cost of my carotenoid, while it is 729 rupees for the commercially available carotenoid.
Based on your basal media.
Based on the statistically optimized media.
Okay, fine. Good presentation. I hope you have very good number of publications also.
Yes,
Congratulations.
thank you.
Okay.
Oh, hello, ma'am.
One
I
small
have one question.
one small correction is there when you write existing years. Yes should be actually capital.
Okay.
Ma'am, I have one question.
Yes, ma'am.
Ma'am, actually, number of research papers are available on the carotenoid production, but its actual applicability in any field is not broadly described by in research paper also. A number of review articles are there only. So, what is
Yes.
your opinion, ma'am, about this?
I think this topic need more research studies. If it is extensively studied on the lab scale level and not in the form of reviews, then the applicability, and if the technology is improved for the carotenoid production, and if that is transferred to the industry, then definitely the industrial carotenoid production can be enhanced. Nowadays, carotenoid production is done with the help of Hematococcus pulvilis, which is an algal strain at a commercial level.
In India, ma'am?
Not in India, it is in China.
In India, means this topic, though this topic have broad application, but in India, it have very poor
research. Yes.
Poor, ma'am.
Yes. I have also gone through such difficulties during my research work. I was not getting good reference for the studies. I have to struggle a lot for getting good references.
Means what I feel, ma'am, number of microorganisms are producing number of products, but actual in market, chemically synthesized products have demand. So what is the problem? Why microorganism we cannot produce such a products that microorganism can be utilized in that area and they use product and it can be marketed? Citric acid produced by organism, but in market actually chemically produced citric acid is available. Vinegar.
That may be due to the economic nature of India, ma'am, because chemically synthesized products are very cheaply available, or they are more cost effective for the buyer, and the microbial products, they are somewhat, or the comparatively they are costly than the chemically synthesized product. So being a microbiologist, I think it is our responsibility to create awareness about the microbially synthesized product among the population, so that we can consume the naturally produced product.
Yes,
then
only... Hello. Hello.
Yes.
Hello. Hello.
Hello. Hello. Yes, sir.
Citric acid is still produced by fermentation in China. There is no chemical method. Still produced by fermentation.
Sir,
We
but in
also
India,
had in India also we had big plant at Surat for citric acid owned by Vadia. But the thing is, we cannot compete with the cost of production.
Yes, sir.
That's the penicillin also. Penicillin manufactured in also. We had earlier 5 bigger plants in India. But now again we are coming with penicillin production in India, like Arvindo at Kakinada. They are coming with 350 KL 10 fermenter. Such a huge facility they are coming. And government has a PLI scheme, production linked incentive scheme. So first 6 years per sale, government will give 10% incentive.
Yes, sir.
So citric acid is, as I said, is produced by fermentation in China. And when China is a huge capacity production, any chemical fermentation. And ascorbic acid, vitamin C, is produced in Baroda plants, Amoli Organics and all. But they get fermentation step, and then they do chemical step here. Penultimate step is fermentation, that they gulonic acid that they take from China. Okay, thank you.
Thank you, sir.
Thank you.
Thank you, sir, for giving this knowledge. But in India, means all the research in microbiology is really poor. As per my knowledge, sir. Okay, thank you, sir.
I think there is no question from participant now. Should we move further?
Yes, sure.
No, this, hello, Dipti, this, this monoclonal antibodies and biosimilars research is going well in India, but they don't disclose outside. That's right. Like Encure, impure alkane,
Yes, sir.
intas. They are working well now. They have sold their product in international market. Biocon is there. See, some help they take, they might take, like clone and all clone. They take from outside sometimes. They develop here then. But India is also working well in research of biosimilar and monoclonal antibodies
Yes,
with
sir.
regulatory, yeah? With regulatory.
Yes, sir.
Yeah.
So please share your contact number and email also, please, sir.
Huh? Hello.
Hello, sir. Please share your contact number also.
Yeah, yeah, okay.
We want to discuss something we can do, sir.
Yeah, yeah.
Thank you, sir.
Thank you, sir.
Okay. Now it's time for thanking our today's speaker. Thank you, ma'am, for your presentation and giving your valuable time. Also, thanks to all participants for attending this webinar and making the time to be with us, and also making this webinar successful. So, thank you all. Thank you.
I am also thankful to Madam, and I am also thankful to Dr. A.M. Deshmukh sir for giving me opportunity to present here and having a great discussion with my PhD topic. Thank you, ma'am.
Okay. Okay. Thank you, ma'am. Okay, ma'am, you may left the meeting.
Yes. So
Okay,
with the
thank you, everyone.
with the permission of today's patron, Dr. A.M. Deshmukh sir, I declare that this session is over. Thank you, sir. Thank you, madam.
Okay, thank you, ma'am.